Hello, this is Tom Seeley. I'm coming to you on St. Patrick's Day 2020 from my bee lab at Cornell University. I'm sorry I cannot be with you in person because of the global pandemic, but I hope you'll enjoy this talk. Title is Darwinian Beekeeping, an Evolutionary Approach to Apiculture. Now, to begin, let's look at who these two gentlemen are. Who's the gentleman on the left and who's the gentleman on the right? Well, probably many of you recognize the gentleman on the left as Lorenzo Langstroth, and the gentleman on the right is Charles Darwin. You can see they're sort of similar in age in these representations. Both have silver hair, some on the top, some more below. And you can see that they were born on almost simultaneously, 1810, 1809. The lifespans were a bit different. The reason I've juxtaposed them is they are both relevant to us in our beekeeping. Langstroth, of course, is the inventor of the movable frame hive, but also Charles Darwin, especially now, for his ideas of evolution by natural selection, which I think have very considerable importance and relevance to beekeeping today, especially as we're dealing with the greater and greater problems of health due to parasites and pathogens of our bees. Now, we think of Langstroth for his movable frame hive, shown here, and we think of Darwin in terms of evolution by natural selection. Darwin was also, though, very much an admirer of comb building. He called it in his book, The Origin of, whatever that book was called, escapes me right now, Origin of Species. He wrote in chapter, I think it's chapter 6 on instincts, he referred to the combs built by honeybees and their comb building as the most wonderful of all instincts. And for him, that was a challenge to understand how evolution could produce such behavior as to build such perfect combs. But he then realized that these cells they achieve their hexagonal perfection by the bees building them together and pushing the cylinders together. You will, like soap bubbles being pressed together, you'll get a nice regular hexagonal array. Now, the reason I'm juxtaposing them, the main reason, is that they both had insights that can help us with our beekeeping today. And for Langstroth, it was, of course, the concept of the movable frame hive. For Darwin, it is, as I've mentioned already, it's the concept of evolution by natural selection. And just to remind us what that concept of natural selection is, we can use an example over here on the left, the evolution of pesticide resistance. This diagram represents individuals that have different levels of resistance to a pesticide, different colors, low level, yellow, very high level of resistance to pesticide, red. And so we start out with a population that's got individuals of a wide range of resistance levels. And that's before selection. Then pesticide is applied, and no surprise, it's only the individuals that are represented by red, which have a high level of resistance to the pesticide, which are still around. And so the final population, as these individuals survive and reproduce, you develop a population where almost everybody has this resistance to the pesticide. So that's one of our challenges in using pesticides today. And and you probably all know that really is a problem. Bugs become resistant to pesticides. So that's the concept of natural selection. Things change by the differential survival and reproduction of individuals based on traits that they have, which have a genetic basis. So How does this concept of natural selection apply to bees and beekeeping, you might be wondering? Well, for one thing, it tells us that everything that colonies do when they're living on their own and they're fully in control of their lives is done to favor their survival and reproduction. That's what natural selection favors: organisms that are able to survive and are able to reproduce. And every little detail of an organism um, has been sculpted, shaped, molded by natural selection to do that. And including every little thing you see with the bees, just the fact of when the bees rear their young larvae, they have them floating in brood food. That's an adaptation for the proper nourishment and care of those young larvae. This bee, all of her traits as she's out foraging, the beautiful um, pollen collecting apparatus, pollen holding, the pollen combs on the hind legs, the beauty of her flight mechanisms, her ability to carry home loads of nectar, her intricate tongue, all of that stuff has been shaped by natural selection for that bee to be an efficient forager. And here again, we've got the honeycombs, the storing up of the honey. That's their winter heating fuel and their heat production. All of these traits, all of those are traits that favor the bees' survival and reproduction. And I think one of the generalizations you can make is looking at bees as products of evolution by natural selection is that the bees are the best beekeepers. They have been molded by millions, truly millions of years of evolution by natural selection to be good at surviving and reproducing. We have fossil records of bees, that beautiful, beautiful fossils. Some of the finest fossils in existence of insect fossils there are of honeybees. Some of these are from shales in Germany, for example. And they go back at least 30 million years. So honeybees have been around a long time. They've been shaped by selection for a long time. And the key idea of Darwinian beekeeping is a simple one. It is the idea that to allow the bees to use their own beekeeping skills fully. Not to second guess the bees, not to presume that we know more about beekeeping than they do, but to let them run their lives as they see fit. Sometimes this is called natural beekeeping, other times apicentric beekeeping or bee-friendly beekeeping. I chose Darwinian beekeeping because I think that name highlights the fact that it's based on the ideas of Charles Darwin. Now, you might be wondering, can we really let the bees be the beekeepers? Haven't all the wild colonies been killed off by Varroa? Let's take a look at that question. We've got our managed colonies, and then we've got our wild colonies. And when I say a wild colony, I just mean a colony that's not being managed. So it might be in a tree, it might be in a building, it might be in an abandoned hive. And so the question is, haven't all those wild colonies died off because nobody's been giving them treatments for Varroa? Aren't they all gone? And the answer is no. As we'll see, wild colonies are thriving in remote places. I don't know in the UK whether wild colonies are thriving for sure, though I have a number of individuals that are reporting to me that they've been tracking colonies living in chimneys and trees and walls, where the colonies, those sites have been continuously occupied for many, many years. And the longest that somebody's reported to me has been about 10 years. So, though I haven't studied the wild colonies in the UK, I suspect they're thriving. What I have studied are wild colonies living in places in North America. But let's recap a broad view across Europe and North America. We have the Arnott Forest in the USA. I'll be sharing a little bit about that. There are wild colonies or unmanaged colonies living on Gotland Island in Sweden, the southern tip of that island. There 40 colonies were taken there and just left alone, and most of them died from Varroa, but there are a number of survivor colonies, and those are repopulating that area. Another area is that I know of is the Chlesch Peninsula. I don't really have pronounced that quite right. It's maybe it'd be better to say northwestern Wales, not just that peninsula. There are beekeepers that have decided to not treat their colonies, and they're finding that their colonies are surviving now well. A wonderful example is the Ile de Groix off the coast of Brittany in France. It's an island, several square miles. There's about 80 colonies, some managed, some living in chimneys in the wild, and nobody's been treating those colonies, and those colonies are doing fine. And then there's evidence for another area in France from forests around the city of Avignon. The study location I know best is the Arnott Forest. This is a forest in New York State, southern New York State. Here we are in North America. New York State is here. New York City is down here. Where I live is in southern New York State in the town of Ithaca. And this Arnott Forest is 17 square kilometers. And it is the one place in North America where we have data on the wild colony abundance before the arrival of Varroa. Varroa arrived here in the early 1990s. It was in my hives by 1994. But the key point is that before Varroa arrived, well before Varroa arrived, back in 1978, I mapped out the wild colonies living in this forest. And this is just a view from the forest from one of the higher hills looking out across the forest in October when the fall colors are at their peak. I found one colony per square kilometer when I searched through this forest. And you might wonder, well, how does one locate wild colonies? You could do it by beelining or bee hunting. You go to a patch of flowers using a little bee box device. You capture bees off the flowers, lure them into, it's a 2-chamber device. You capture them in the box, you lure them back into one of the chambers, and then once you've got a bunch of bees, maybe half a dozen bees in the rear chamber, you open up the front chamber, you put in a little square of comb with sugar water, then you let the bees come forward from the rear chamber to the front chamber. They find the comb. They think it's great. Unless there's a honey flow on, they will go home, bring their hive mates, and you'll develop a traffic of bees between your comb and their home. And then you work your way down that. You can see which direction they're going. You can time the bees to get a sense of the distance. But step by step, you work your way back to their home. Here's an example of a bee hunt that I did on the Cornell campus, where I started with bees at a flower garden here. I could quickly see, it was early spring, the bees were quite hungry. There weren't many flowers out yet, so the bee lining went very quickly. The bees were excited by the comb, and they quickly showed me that they were flying home. I thought at first they were flying off in this direction, so I moved after about an hour where I've got the line established. The bees' interest had built up. I moved my feeding station to the next red triangle here. Then it became very clear they were going due north, so I could, over the next couple of hours, I moved to here, then to here, then to here. 4 o'clock I got here. It was very clear that the bees were close by. Their home was nearby because it was only taking them 2 minutes to leave that location, to go back to their bee tree and then reappear at my feeder. So I searched down this line. There's a little forest area here. I searched there and I found bees flying. Could see bees flying in and out of this oak tree, red oak there. And they were right from that, where that red arrow is. Let's see. Okay. Now, in the Arnott Forest, so that's how you go bee hunting. In the Arnott Forest, in 2002, I went, which was well after the Varroa had arrived, I went back to the Arnott Forest and I lined the bee trees in the Arnott Forest. I started in the back corner. I got a line to one bee tree here, found that tree, got a line going to another bee tree up here, found that bee tree. Then I started at another patch of flowers, got one bee line, went back to the tree that I had found already. The other line had gone up to a new bee tree, found that, and started with another bunch of bees. One line pointed back to one of the trees I had found. One pointed to a new tree. So just bit by bit, I worked my way around from clearing to clearing, from various clearings in the forest, and I found, looks like 8 bee trees in 2002. So, and as I want to stress, Varroa had been in my area in New York State since the early 1990s. So the bees were still alive even after 10 years of Varroa being in the area. And we now know that this density of colonies that I found in 2002 is the same density as I found back in 1978 before Varroa arrived. So the bees are still present in the wild. And I know that it's not, they're surviving is not simply because, is not because they lack Varroa. All of I put up bait hives in the Arnaud forest to capture swarms moving into those bait hives, and then once they're moved into the movable frame hives, then I can measure the Varroa, and I can check for other diseases as well. And you can see that all 11 colonies that I caught in my bait hives had Varroa. So all of these are well equipped with Varroa, you might say. One of the colonies had chalk brood. None of the colonies had, however, American foul brood or European foul brood. So key conclusion, wild colonies in this Arnott forest in 2002 were infested with Varroa, and yet they were surviving. And here's a way to look at this. This is based on, this slide illustrates how a population of wild colonies, how this population of wild colonies persists. And this is based on data I collected in 2010 to 2017, where I'm just checking colonies, nest sites that are occupied. And here's the logic of how it works. Let's say you've got 100 locations where bees are living in a tree or a building. And those would be, and you know that they're there from the previous fall. So those would be what I would call established colonies. So let's say you have, in May of any given year, you've got 100 locations occupied by established colonies. Well, between May and September, what's going to happen? Well, almost these probabilities, these fractions, 0.97, that represents the survivorship. So of those 100 colonies, sites that are still occupied, 97 will still be occupied in September. So there's very little mortality of these established colonies over the summer. These established colonies will also be casting off swarms. I know from my investigations that the probability of swarming by an unmanaged colony living in a cavity of the size that these wild colonies live is about 0.87. So by September, in addition to the 97 locations of the 100 originally that are housing a colony, there's another 87 locations housing a colony elsewhere based on the swarms that were cast off from these original locations. So you end up with 184 sites occupied by wild colonies. And 87 of them are what I call founder colonies, and 97 are what I call established colonies. Now, what happens over winter? Well, those established colonies have a mortality rate of about 16%, or 0.84 survival, probability of survival. So you end up with 81 of those 97 are alive next May. And the founder colonies, their survival is much lower because they had a much more difficult situation of moving into a new nest cavity, building columns, rearing brood, storing up enough food. And where I live, at least, only about a quarter of those newly founded colonies survive to the following spring. But when you go through the numbers, you see that there's a loss of, the losses from the established colonies are almost perfectly balanced out by the additions of the following May of the founder colony. So there's 81 of the established colonies still alive in May, but there's 20 of the founder colonies still alive in May. So that brings the population back to 101. So that was our starting point in May of this year, 100 established colonies, and on average, we will end up with 101 or 100, whatever, close enough, uh, colonies the following May. So basically what's going on in the wild is colonies are swarming a lot. Those established colonies have pretty good survival. Founder colonies, there's lots of them, but they have low survival, and everything, there's the founders contribute, their survival matches the losses of the deaths of the established colony. So each winter, 19 of the established wild colonies die, and about 19 of the founder colonies survive. That's how this wild population of wild colonies persists, at least where I study them. And one thing I want to stress is that I don't think there's anything magical about this forest. It's just an example of the right conditions. And we now know that there's some other forests near where I live, where this same pattern is going on. And there's also been a study being done, it hasn't been published yet, in Pennsylvania, where the same pattern has been found. The wild colonies are there, they swarm a lot, and the swarm-based colonies or founder colonies balance out the mortality of the established colonies over winter. So with that as background, let's look at what I mean by Darwinian beekeeping or apicentric beekeeping. How does it work? What are the ideas here? One thing I want to stress at the beginning is that I don't have all the answers on this yet. It's a work in progress. A lot of people are working on this, how we can adjust our beekeeping to make it more bee-friendly. And a couple of things I want to stress. Another thing I want to stress at the beginning, it's not for this Darwinian beekeeping is not for large-scale beekeepers. It's not for urban beekeepers. And what it is, it's an option for small-scale rural beekeepers who want to avoid chemical treatments of their bees and who are satisfied with modest honey crops, about 15 kilograms per hive or thereabouts. So that's what it is. It's not your traditional kind of beekeeping where the emphasis is on production, honey production, or crop pollination. It doesn't involve typically the Darwinian beekeeper will have just a handful of colonies even, so they can be widely spaced. Now, to hammer home or emphasize this difference between Darwinian and commercial beekeeping, I'll draw some parallels. To my mind, Darwinian is to commercial beekeeping as bird watching is to poultry farming. Bird watching, we go out, we do it just for the sheer enjoyment of watching the birds. We don't expect the birds to give us eggs or meat or anything like that. Poultry farming is very different. You don't expect to have a lot of enjoyment from poultry farming unless it's on a small scale. But you do expect to get some eggs and maybe some meat from your birds. So another analogy or a way to illustrate the difference is the difference between having one backyard apple tree versus a commercial orchard. You can imagine the one backyard tree apple tree is like one backyard hive. The commercial orchard is like a vast apiary. And another contrast is one is about pleasure and the other is about profit. So this Darwinian versus commercial beekeeping, they're very different approaches to beekeeping. And I think I'll maybe want to reinforce this key point about understanding about how Darwinian beekeeping works. It is that to draw this distinction between the bees' original environment, the environment in which the wild colonies have lived for millions of years, and in which some wild colonies can still live. And the other situation for bees in their living are what we'll call current circumstances. And this is the environment in which most colonies live, the managed colonies live. And the way what we want to do with Darwinian beekeeping is we want to see, okay, how can we have bees living in our beehives, but put them into a lifestyle or environment that is like what wild colonies experience, but is not what managed colonies experience. So our goal is to put, in Darwinian beekeeping, is to put managed colonies, insofar as possible, back into the honeybee's original environment. And we're going to go through this step by step. And I want to stress again at the beginning, I don't have all of the answers. I think I have some of the answers, but it's very much a work in progress. And there's more and more papers, investigations being reported on this topic all the time. I'm making a collection of these, and I'm getting more information. And in a year or two or three, I hope to be able to write a more complete description of what we're learning about how to make Darwinian beekeeping work. But let's go through some of the key things that I believe we understand already. One difference, and we're going to go through it as a matter of making comparisons. A comparison of the wild colonies and managed colonies. Well, the original environment, the colonies are genetically adapted to their location. Current circumstances, often the colonies are not genetically adapted to their location. Why is that? Well, beekeepers, especially in North America, but I know it happens here too in Europe, they ship queens all over. And so we've got, for example, you can buy in North America, you buy Hawaiian queen bees from the Hawaiian Queen Company. And these are the ultimate queen bee from Hawaii. They may be the ultimate queen bee for Hawaii, from Hawaii and for Hawaii, but I don't think they're the ultimate queen bee for northern climate regions. But just the fact that we ship them around between continents and things like that, it illustrates just how much mismatch we can create, with our colonies between their level of adaptation to their current location. Here's another contrast between original environment and current living circumstances. Originally, colonies lived widely spaced in forests. Now they often live crowded in apiaries. There's a bee tree, one of the bee trees I found, and this is one of my bee yards, one of my apiaries. Another contrast is the size of their homes, their dwelling places. In the wild, this is a distribution of the nest cavity volumes of bee trees that I've collected. You can see they're generally in the range of one deep 10-frame hive body, sometimes a little smaller, sometimes a bit bigger, but they're not 2 deeps, and they're certainly not 3 deeps. In contrast, current circumstances, well, this is a photograph that I've lifted from one of the bee supply catalogs here in the US. And you can see that these bees are living in, this is the best and most complete wooden kit, by the way. You can see standard beekeeping practice, you put bees in very large hives, supersized nest cavities, multi-storied hives. In nature, they would live in this lower hive box, this one box at the bottom, basically. And so all of this stuff up above, what does it do? It gives the bees lots of space to build up, have lots of brood, and store up lots of honey. And not swarm, rarely swarm. And these, when they're living in a small hive like this, they don't store up a big surplus of food, and they swarm freely. Big difference, fundamental difference. Another contrast between original environment and current circumstances is that in the original environment, in nature, they coat the walls of their nest cavities with propolis. And why are they doing it? Well, it's because those inner wall surfaces have been produced by, are riddled with crevices and cracks from the fungal decay that produce the cavity. That's when those surfaces like that, which are spongy and impenetrable to the bees, when those get wet, it's a perfect place for mold and microbes, other microbes to grow. So the bees seal that off with propolis. And that propolis, in our hive walls, in hives, the walls are not coated with propolis. You might say, well, they probably don't need it. And yes, from the bees' perspective, they don't need it. But there's other effects of this propolis besides its physical sealing off of cracks and crevices. It has antibiotic compounds in it, which help the bees, and we'll talk a long time about that. But so these bees with the propolis are enjoying protection from microbial infections in ways that bees do not enjoy when they live in our hives because they haven't coated the walls with propolis. Another contrast is in the wild. Colonies build drone comb freely. In the nests, wild nests that I've dissected, it was in the range of 15 to 20% drone comb of the comb area in the nest was devoted to drone comb. In our managed colonies, we generally discourage bees from building drone comb, though some beekeepers are putting in a frame with drone foundation or just no foundation to help the colonies produce drones. But in general, under beekeeping management, colonies are discouraged from producing drones. And one of the consequences of that is that if you've got colonies with good genetics, managed colonies don't share those genetics, that don't share their genes as effectively as they would if they had drone comb as in the wild. Another difference, really big difference, is in the wild, at least where I live, the nest entrance is usually high off the ground, an average of 8 meters up. Now this one, here's a bee tree I found, that's me, 2 meters approximately, and then here's the nest entrance of this tree, way, 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 way up here, very high up. Whereas our managed colonies, the entrances are low to the ground. Now, we don't fully understand what the consequence of that is, but these bees definitely, if you give them a choice, they'll always go for a high nest entrance. Some of that might have to do with making their nests less conspicuous to predators, particularly black bears in the forests where I work. But it may also have to do with the fact that these higher sites in spring, coming out of winter, these are probably sunnier and warmer than places that are down close to the forest floor. And that's one of the many unknowns. But I do know that when bees in the winter fly out from up here, they don't crash into the snow. Whereas I know from my bees, when I've got my hive, where my hives are, of course, sitting on the ground, or bees often come out, get a little, their flight's not real strong. They'll crash onto the snow, and then they're toast. They're gone. They can't warm up enough to fly back. So another big difference: high versus low nest entrance. In original environment, the colonies have diverse pollen sources, but in many situations, in current circumstance, the colonies sometimes have non-diverse pollen sources. And the extreme of that, of course, are the vast almond orchards in California, where they have just one pollen source. Another contrast is that in the wild, colonies are not treated for diseases, and so the bees evolve resistance to their diseases, including varroa. Whereas in management, current circumstances, colonies are treated for diseases. And that might seem good it is probably good for the bees in the short term, but it also means that it blunts selection for resistance to the causative agents of disease, such as varroa. So, back to the idea of Darwinian beekeeping. Insofar as possible, put managed colonies back in the bees' original environment, their environment of evolutionary adaptiveness, to use the fancy jargon. But in essence, the idea is to let managed colonies live more naturally. The colonies will certainly make less honey, but they will enjoy better health. So it's a production versus health trade-off, and that's much as it is in agriculture. You get high production, but it's a lot harder to keep the plants and animals healthy. How can you do this? So what are some of the ideas for this Darwinian beekeeping? One is, and it's fundamental, keep bees that are adapted to your location. Now, how can you do that? Well, it's not that hard. You can rear queens for your best survivor colonies, or capture swarms with bait hives, particularly if they're put in remote locations away from beekeepers. You'll tap into the native stock, the local stock, or you can purchase queens from a queen breeder who produces locally adapted queens, somebody nearby you. just a little bit. I get my bees using the first technique. I put out bait hives for catching wild swarms. My occupation rate varies from year to year. Sometimes it's 20%, sometimes it's over 100%. Average is 58%. And this is just one of my bait hives. It's a 6-frame nuke box, as you can see. I've left it on top of an old hive, not one of mine, but one of a beekeeper nearby. I put it there, and then I actually forgot about this one. So the bees moved in one summer. I came back the next spring, and there the bees were alive, even though it's a pitifully thin-walled box. They were alive, and I took out the bees. And this is always a wonderful sight. You find the queen, and that queen is, you don't know where she's from, but she's produced a beautiful colony that survived the winter all on her own, just in this box. Tough bees, excellent bees. And one thing I want to stress is the bees that I get are gentle bees. They're not really mean bees. Every once in a while you'll get a colony that's a little touchy, but for the most part, they're quite nice bees. They make strong colonies. No chalk brood. Have never seen any chalk brood in the colonies that I've caught. And they have very good varroa resistance, all on their own. They don't need treating. I'll show you that in a second here. Oh yeah, here we go. In summer of 2019, I did a small experiment where I set up 3 groups of colonies, and I set all these colonies in each 3 group in the same location. They're all started as 2-frame nukes on the 19th of June, 2019. But the way the colonies of the 3 groups differed is where their queen came from. So in one group, the colonies were set up and they were given queens from a queen breeder in California called Oliveris. And they produce lots of what they call Italian queens. The second group, the queens came from swarms they caught with bait hives earlier that spring. And then the third group, the queens came from a non-treatment beekeeper called named Kirk Webster up in Vermont. And so the colonies were all set up the same way, same place. They weren't, the hives weren't mixed together. There were 3 small apiaries along the same tree hedgerow with about 50 meters between them. And on the 9th of October, 2019, I came back and I measured the mites per 300 bees, a standard sampling technique using the sugar shape technique. And you can see that for the Oliveris, California queen colonies, the mite counts were pretty high. My bait hives, the mite counts were low except for 1 colony. And the Webster colonies, the mite counts were rock bottom, very, very, very low. So I expect, I guess I don't have it a slide here, but I expect, well, actually I can tell you now because I just checked the colonies last week. Of these Oliveris queen colonies, there are 2 that are still alive. This one and this one, where the colonies had low mite counts. The colonies with my bait hives, they're all alive except for this one. It had lots of mites. It died. And the Webster colonies, they're all alive. So these are almost all dead. These are all alive. And these are almost all alive. So it's a good example of getting the effectiveness of getting queens that you've caught from the wild in your area. So second suggestion for Darwinian beekeeping, house your colonies in small hives. And when I say a small hive, what I'm referring to is 1 deep 10-frame hive body and 1 shallow or medium honey super over a queen excluder. And the reason you want to use a queen excluder is you want to be able to take that honey super off without removing brood. If you want to take honey away from the colony, you don't want to mess up their brood nest. So, and I don't really have time to go through it, but and as I say, this is a work in progress, but I'm trying to figure out, I think a key thing will be if you want to take honey off, if you want to put on a shallow super, you do it and you take the honey super off early in the summer so that you give the bees most of the summer to refill, to fill up their hive with honey. And I'll just share with you the difference between the power of putting colonies in small hives. I did one experiment where I set up 12 colonies, and in one group of the set up two groups of 12 colonies. In one group, the colonies were installed in small hives. The other group, they were installed in large hives, and both groups were set up at the same time. And in fact, the colonies were the queens all came from a queen breeder, and they were same age queens and so forth. And these colonies in the large hives, they were set up in the form that honey managed for honey production. This would be the typical four deep hive bodies, 2 for a brood chamber and 2 for honey supers. And these were just left in 1 deep hive body. And these colonies were given no varroa treatments, and all of the colonies survived their first winter because all the colonies started out as small 2-frame nukes. And so they got through the first winter because their varroa counts were low. But the second summer, the colonies were well established, and at the end of the 2 year, after the end of the second summer, only 17% of these colonies were still alive. Again, no varroa treatments, but 83% of these were still alive. The difference is small hives, these colonies swarmed, they had breaks in the brood rearing. They became honey-bound early on in the late summer, so that shut off the brood for the mites to reproduce. And big impact of hive size. Here's an example of this is the numbers of some of these mite counts. The colonies were set up in June, but a mite count was made in July 2012, and the mite counts were very low, mites per 300 bees. Next spring, the mite counts were again very low, but in the large hives, the mite counts went up and up and up, and then the colonies were crashing in the fall. In the small hive colonies, the mite counts stayed low, and thus they survived. And this is all without varroa mite treatment, so 80% survival in the small hive treatment without mite treatments. The third suggestion for Darwinian beekeeping is to space your colonies as widely as possible, and did an experiment to look at the impact of this. Set up 12 colonies again, all of them put together in an apiary situation. And then I had another 12 colonies, which were paired with these 12, but they were nearby, but they were not crowded together in an apiary. And these colonies were set up at the same time, in the same place, same 2 deep hive body hives in this case, and none of these colonies received a varroa treatment. And what we found is there was less drift between the colonies in this situation. I don't have time to go through it, but we used color markers so we could look for the drifting of drones. We found less spread of mites between colonies in the dispersed situation, and better survival of the colonies. And I want to stress, these colonies were not given any varroa treatments, and they're all headed by queens from a queen breeder, Oliveras in California. And so I think the next slide shows, the survival. After 2 years, these are all gone, all dead. And 5 of these were alive after 2 years, which was a little surprising to me, given the source of the queens from the California queen breeder. But 5 of them had the ability to live on their own. And I think, but what happened over here was that there was a very strong mite bomb phenomenon among these colonies. As soon as some of them started to collapse, they would be robbed out by robbers from nearby colonies, and so the mite counts shot up in all of them. Here there was a little bit of that mite bomb, a little bit of robbing, but it didn't happen as much, and so there wasn't as much spread of the mites in this situation. The 4th suggestion is to line your hives with propolis collection screens, or build the hives of unfinished lumber. Either way, the point is you're trying to encourage the bees to deposit propolis on the walls. And I don't know if they have these in England, but in North America, you can buy these propolis collection screens. They're just plastic screens that have slits in them, slots in them, and the bees fill those with propolis. You can use those encourage propolis collection on the walls of your hives just by stapling that material to the walls. And this is what it will look like after you peel this plastic propolis collection screen material off. Lots of propolis has been put on the wall. It creates an antimicrobial shroud around the bees. Fifth suggestion, provide your most resilient, your lowest mite count colonies with 10 to 20% drone comb. And the logic here is you want to promote the genetic success of your best colony. So all those drones you see that colonies are making, if it's a good colony, you want that colony to be spreading its genes. And you can help it do so by enabling it to, by giving it lots of drone comb to enable it to produce lots of drones. Sixth suggestion, keep nest structure intact. Put each frame back in its original location and with its original orientation. No reversing of brood boxes, etc., etc. The bees work very hard to organize their nests, and probably the last thing we want to do is disrupt the organization of their nests. That must be very, very disruptive to a colony. And I suggest 7, provide only a small entrance at the bottom of the hive. And you might wonder, why is that? Well, we know that, first of all, the bees don't like a top entrance. Probably those of you that are beekeepers, all of you presumably, you know that the bees don't want a top entrance. They're in the fall, they're sealing off your inner cover, they're plugging top openings with propolis. So you don't want to give them a small entrance at the top, and that's for thermoregulation, reasons of thermoregulation. Instead, what you want to do is give them a small entrance at the bottom of the hive and add a styrofoam board above the inner cover, because you want to insulate, give them good insulation above. And what I do in my hives, I give them just a 2-inch wide opening, and then I put 1/2-inch mesh screen to function as a mouse guard over the opening. And I want to look more carefully or more precisely at why you want to avoid a top entrance and why the bees want to avoid a top entrance. It has to do, and here I'm going to refer to work by Derek Mitchell, where he's described how if you've got an insulated hive, a well-insulated hive, and if there's no top entrance, then there's a hot pocket or layer at the top of the hive where the bees will be overwinter very comfortably and easily with low honey consumption. But if you put a top vent in the hive, then you've created, and then the air, you've got a perfect convection current situation where warm air is going out the top and cold air is coming in the bottom. And so you have a much smaller area of warm air in the top of the nest. And I know a lot of people argue that, we should give them a top entrance to minimize condensation. But what Derek Mitchell is finding is that if the hive is warm, if the upper combs are warm, then there's not condensation because the surfaces are not below, their temperature is above the dew point. What condensation will occur in a hive in this situation will be the lower combs away from the bees. So so the bees will not get wet from their own condensation without a top entrance. And I've reproduced this with my own colonies now, and it's exactly what I see. I don't put a top entrance, but I insulate the top, and I open the colonies in the spring, and there's no moisture, there's no mold. The bees look really good. And I know that the bees, from other studies we've done, we know that the bees do need water in the winter, and so having some condensation in the hive, just not above the bees, is actually beneficial to them. They need water and when you think about it, that's not surprising because they're living off of honey, and honey is an 82% sugar solution. And I think we all can empathize with what it would be like if you couldn't drink any water, if you were eating just honey. You would have osmotic problems. So one of the most important things we really have can do is to improve the insulation of our hives. It's one of the something we've only now are fully understanding why that is. And I think this is my last suggestion. Do not disturb the colonies in the winter. No checking, no stimulative feeding, no pollen patties. Because we know that some work I've done and others that even a brief removal of the nest lid, the hive lid, causes the winter cluster to raise its temperature in alarm for several hours. And who knows what else it's doing to the bees. They're best left alone, I think. Oh, and last one, this is a hugely important one. Refrain from treating colonies for varroa. It's also a very controversial one. It does not, however, say that you shouldn't be monitoring the varroa. You must monitor the mite levels, I feel, the mites per 100 bees. And if the mite level gets too high, more than 10 per 100 bees, or maybe you could even use more than 5 per 100 bees, or even 2 per 100 bees, then I would euthanize the colony, because that colony is showing you that it doesn't have good resistance to the varroa. And the last thing you want to do is perpetuate that colony. You don't want it to be surviving, making drones, casting swarms, etc. And the way to do that is to euthanize the colony. You can or you could requeen the colony. If you want to euthanize it, I would advocate pouring warm, soapy water into the hive at dusk. It eliminates your non-resistant colonies, and it avoids, equally importantly, it means that this colony will not collapse and turn into a mite bomb. And by a mite bomb, I mean a colony that has lots of mites, is getting very weak, gets robbed out, and so it spreads mites to lots of other colonies. Mite bomb is probably not the ideal term for this kind of colony. I'd call it a robber lure, but mite bomb seems to be what people say. And as I say, an alternative to euthanasia is to treat the colony for varroa and requeen it with a queen of resistant stock. Now you might be wondering, well, what kind of colony survival am I going to get with this Darwinian beekeeping? Well, I've tested that. I've done it by just putting out colonies using this Darwinian beekeeping, though I haven't yet used well-insulated hives. So these results are probably not as good as you would get if you had better insulated hives. I just put out colonies in a 10-frame deep hive body, styrofoam insulation above, small entrance, etc. I put them around the countryside. I set up 10 or 12 colonies per year, so I spaced them out. And what I found was that my summer survival was 97%. And I'd set up these colonies by capturing swarms. I would capture swarms in bait hives and then transfer them into these hives. So they had locally adapted queens. Summer survival, 97%. Winter survival, 84%. So overall survival over those 7 years, or actually 8 years, was 82%. And you might be thinking, well, this Darwinian beekeeping works well in Ithaca, New York, because it's a place with no commercial beekeeping, and it has forests with lots of wild colonies. And it's true, it also is a place where natural selection had created a wild population of varroa-resistant colonies. But you don't need a small town situation like Ithaca, New York, with forest lands all around. You can also foster natural selection to get varroa-resistant colonies. And here I'm going to refer you to a paper that's come out of the Netherlands, called Darwinian Black Box Selection for Resistance to Settled Invasive Varroa Parasites. And basically what they're saying is all you do is they call it Darwinian black box because you're not sure what's going on in the colony. The colony is treated as a black box. But what you are doing is you're taking a colony and you're splitting it up. Well, I encourage you to go back to this. You would just want to Google Darwinian black box selection, and that will take you through this process. It's basically a process where you set up a bunch of colonies, you see which ones survive, which ones are live. Then you split those colonies up into other colonies, make splits, set them up, and then Um, let them rear their own queens, and let them survive. And you just keep going through that process. And you just every year you let the ones that don't survive, you let them die. You don't coddle them. And bit by bit, over it doesn't take very long. You've got colonies that have resistance to varroa. So it starts with 20 to 30 genetically diverse colonies, a remote location or a moderately remote location. So queens will mate within the population. No varroa control, and probably takes about 4 years. Go back to that. After 4 years, you have given time for natural selection to work to give you colonies that have good resistance to varroa. You're not breeding, you're just letting natural selection do the selection, not artificial selection, just natural selection. I hope this has been useful to you. Thank you very much for your interest.